Well before you take your first breath, you are already a product of your genetics. When your genetic blueprint fails to perform its intended function, this can lead to serious and life-threatening conditions. Advances in stem cell research have allowed scientists to further understand the mechanisms that cause such conditions and allow the postulation for potential treatments. So what exactly are stem cells, and how can they be used to treat genetic diseases? All cells in the human body have three things in common: a nucleus, cytoplasm, and a cell membrane. Stem cells are no exception, but what makes these unspecialized cells unique is two key properties. Firstly, they have the ability to self-renew. Secondly, the ability to differentiate into specialized cells such as skin, lung, or cardiac cells. This allows stem cells to serve as the body's repair mechanism. This process of cell specialization is called cell differentiation and occurs when a cell selectively activates or inactivates specific genes to enable specialized cell function. There are 5 different types of stem cells that are found at different stages of life, in different locations in the body, and range in their differentiation capacity. Topipotent stem cells are the first cells of life and occur in the first hours after fertilization. These cells are able to specialize into all cells within the body and can form the umbilical cord and placental tissue. Pluripotent stem cells occur in the blastocyte, an early embryonic stage, and can form into any cell within the body. Multipotent stem cells occur in the adult stages of life and can only change into cells of that specific organ. Oligopotent stem cells have the ability to specialize only into a few select cells, such as particular muscle cells and a few types of blood cells. Unipotent stem cells have the ability to specialize into only one type of cell, and at this point, still a lot of research is needed to be done. Research into embryonic stem cells dates back to the mid-1970s. Following successful bone marrow transplants and in vitro fertilization, it wasn't long before blood stem cells were discovered in umbilical cord tissue. In 1981, Martin Evans of Cardiff University was able to derive embryonic stem cells in mice. Over the next decade, stem cell lines were created in other animals, accumulating and cloning of the dolly to sheep. In 1998, James Thomas and John Gerhardt were both able to develop pluripotent stem cells. Thomas by isolating and growing stem cells from human IVF embryos, while Gerhardt derived cells from fetal gonad tissue. For all stem cells, environmental factors influence the type and rate of differentiation. Chemical and physical signals from the environment produce a response in the stem cell. This influences gene activity and in turn leads to cellular differentiation. Researchers aim to replicate these environmental conditions ex vivo in order to replicate the differentiation of the stem cells. They use techniques such as signal inhibitors, growth factors, and co-culture environments to manipulate the differentiation of stem cells into a specific cell type. In Austria, between 2004 and 2011, 9 children took part in a trial that tested the use of hematopoietic stem cell transplantation to increase the production of normal red blood cells. The children had sickle cell blood disease, a genetic condition that leads to the abnormal formation of sickle-shaped red blood cells, reducing their ability to carry oxygen. The procedure involved collecting the multipotent stem cells from the siblings' bone marrow that doesn't have sickle cell anemia, and depositing the functioning cells into the patient's bone marrow. The success of the trial was apparent after just a 4-year follow-up that indicated that all patients were disease-free and the implanted stem cells had continued to produce normal red blood cells. In order to conduct research on embryonic stem cells in Australia, a licence must be granted by the NHMRC Embryonic Licensing Committee. The research proposal must be assessed and in compliance with the National Statement on Ethical Conduct in Research of Human and following Assisted Reproductive Technology guidelines. These policies were introduced to help safeguard the public from unethical stem cell research. Due to restrictions in research, there are very few options in Australia and many people choose to travel overseas for stem cell therapy. Despite the risk, desperate patients volunteer to take part in radical experimental treatments with the hope of finding a cure. This can lead to harmful outcomes as the treatments are often not scientifically sound. Health risks have included infection, immune system rejection, and cancer. Stem cell research has led to many promising discoveries, such as the work of scientist Shinya Yamanaka. Yamanaka's team was successful in reversing a fibroblast cell, a cell found in the connective tissue which produces collagen, back into the pluripotent stem cell. This cell found naturally in the blastocyte during development. These cells are now known as IPS, i.e. induced pluripotent stem cells. The discovery means that one day human embryonic stem cells may not be required for research, and instead stem cells could be produced in much larger quantities. As we move on into this new and exciting area of research, we'll need to decide collectively as a society how we will navigate through these ethical ideas about what life is and to what extent we can alter it. This will be tasked to the thinkers and scientists of the future. Thank you very much for listening to our presentation. We're Group A5.